welcome to the channel. So tomorrow is my examination of cell biology, and right now I am revising my sheets whatever I make in the classrooms. Okay, so I'm going to revise in front of the camera. It basically for me, but if you are watching, then it would be fruitful for you because it is a cell biology session we're going to have, and especially for myself I'm recording this. Maybe I uh, make the video private only, but if I make it public for the uses of my friends, then it would be definitely helpful for you because I was revising. Okay, so as my examination was tomorrow, so I'm going to just read the topics. I'm not going to into the clarification of the topic. Okay, and I'm going to start from the very initial. So these are the basics, and I think I've already uploaded. And the topics, definitely the subject was cell biology, and the topics are mainly the basic topics of the cell, and then we have signal transduction, ECM, extracellular matrix, cytoskeleton, and all of these maybe in second or third video, because I'm not going to make the video more than of 1 hour. Okay, per session I'm going to give 1 hour. Okay, so I'm going to start from the very first class of my university. That is the first class, which is happened in 7th of January 2026 by Professor Dr. Gunjan. So classification of organisms. So the um, what are the classification of organism based on the cell structure? So there are, I think, 8 points. So classification of organism based on the cell structure. First one is complexity. The complexity of the cell, whether it is um, prokaryotic, eukaryotic. That is the advance. If we talk about the complexity, functions it performs, evolution, genetic makeup, morphology, anatomy, cellular structure, and reproduction. So these are the classification of organism based on the cell structure. So as we already know, two types of organism broadly classify: prokaryotes and eukaryotes. So the prokaryotes, simple, devoid of true nucleus. So they do have nucleus, but they are not the true nucleus. Okay, so they are devoid of true nucleus. They instead have nucleoid. Okay, it means they do have nucleus kind of stuff, but it is membraneless. It means it is in the direct contact of cytoplasm, and that is also why the transcription and translation is coupled process in prokaryotic and the lower organisms. Okay, because they do not have the membrane. Okay, the second one is eukaryotes, which is obviously the complex one, and they do have a true nucleus present. It means they do have membrane. So difference, what are the differences between the prokaryote and you? These are the simple basic knowledge you must have. They are not going to ask in your examination because uh, these are just for the basics. Okay, to give a start to a subjects, because there are a lot of students who coming from the weaker educational background, so they must have something, some clarity about what is cell and all. Okay, so size, if we talk about size, prokaryotes generally have less size, which is roughly around 1 to 10 micrometer, and the size of eukaryotes is 10 to 100 micrometer. So the size of prokaryote is 1 to 10 micrometer, and the size of eukaryote, which is larger and complex, it is 10 to 100 micrometer per cell. That is the size of per cell. Okay, cell wall, if we talk about, in prokaryotes we have peptidoglycan, which is the repeating units of NAG and NAM. And if we talk about the cell wall in eukaryotes, we have chitinous and cellulosic in plants and chitinous in fungi. And if we talk about the ribosome, so prokaryotes have smaller ribosome, which is 70s, 40 and 30, I think. Yeah, 50 and 30, I think, like that. Okay, and then we have a large ribosome, which is 80s in the eukaryotics. But eukaryote do also have 70s ribosome. Do not forget that, because according to the endosymbiont theory, the the mitochondria and chloroplast are used to be a prokaryotic cells. They somehow get symbiosis with the native cells that provide the protection, and instead of that they give energy and whatever. So eukaryotes do also have 70s ribosomes. Okay, so they have both, but primarily they have 18s as Sedberg unit, Sedberg coefficient. Prokaryotes are unicellular. Eukaryotes could be a unicellular plus multicellular. It means they can be both. For example, plants, animals, fungi. No membrane bound organelle. So there is no membranous bound organelle present in the prokaryotics, and that is the very defining property of prokaryotes, that they do not have membrane bound organelle. But if we talk about eukaryotes, yeah, they do have membrane bound organelle, which helps giving the property of compartmentalization, and thus the division of liver is possible within the eukaryotic cells. True organelle absent in prokaryotes, and yeah, organelle are present in eukaryotes. Now we see the what are the common evolutionary things which keep intact in both of the organism. So the DNA, yeah, both can have it. Ribosomes, both can have it. The ribosomes is the only one that is membrane bound membrane less organelle which is present in both, but membrane bound organelle is not present in prokaryotes. Cell membrane, yeah, which is universal, and cytoplasm, you can say a cytosol area, that is present in both. So you can make diagrams like this. Okay, so it can cover pages, and it is easy for the invigilator to check. Okay, now we see the prokaryotic organism. These are basic, no one going to ask. Okay, so prokaryotic organism, lack of true nucleus. So the prokaryotic organism don't have a true nucleus. I have already talked about it. Genetic materials not enclosed by nuclear membrane. So what does it mean by not having a true nucleus? What does it mean? So it means that the genetic material, whether it is a DNA and RNA, not both, whether it is DNA and RNA, not enclosed by a nuclear envelope, nuclear membrane. There is no protective layer which provide the compartmentalization ability, and thus we have no division of liver in the prokaryotic organism. So they do have a nucleoid instead of nucleus, which is suspended in continuous with the cytoplasm and the cytosolic area itself. Then we talk about the ribosomes, which is 70s. Yeah, indeed. A division, if we talk about the division, if you don't know and someone asks, division about the how they divide prokaryotes, they do not have mitotic and meiotic division kind of stuff, because meiosis for the reproductive, sexually reproducing organisms. So the division by binary fission, they divide by binary fission, not by mitosis. For example, the ribosomes. Okay, so the thing is that they are smaller, so the biotic potential of these organisms are very high, the evolutionary rate are very high, thus their stability is decreased in such organism. For example, the bacteria which are present in diverse environment. So these are the micro question, 0.25 marks, not even 1 marks. So they can give, bacteria as cosmopolitan, bacteria which are present in diverse environment. So this is not going to be question. The question is going to be why. Okay, so for example, the archaebacteria. Okay, so present in hot spring and salt lake, the extreme conditions. Why? That would be a question. Why? So membrane composition of the bacteria is different. Okay, with the others. So the why the archaebacteria survive in such a extreme condition? So it's all because of the membrane composition of their universally plasma membrane. So there would be a difference. Okay, difference in the lipids. And prokaryotes do not have a cholesterol in their plasma membrane. They do have hopinoids. So there must be a difference in the composition of hopinoids and all. So this could be a question. This could be a question. Why? So the simple answer could be due to the membrane composition. Okay. they may be beneficial or pathogenic. So it's not that the bacteria is harmful. Okay, it's not like that. They are beneficial also. For example, lactobacillus, if you recall in your class 12 microbe chapter. So lactobacillus, which is beneficial for the production of vitamin B12, and also they check the quality of your gut. Okay, pathogenic are also be there. Okay, there are a lot of bacterial diseases that can be only treated with the possible treatment is antibiotics. For example, mycobacterium tuberculosis, very dangerous. 8 Jan. Now we shifted to 8 Jan. These are my notes. So compartment, prokaryote, eukaryote, cell concept, two phase concept and three phase concept. So in the cell concept we have taught two concept. Okay, so the first one is two phase concept, the second one is three phase concept. The question would be from the three phase concept, and the question marking would be 2 marks. Okay, so UFM notes according to national level exam. These are the some philosophies by the professor I have noted in my sheet. So cell concept we start. The question could be framed for define cell concept. What are the concept of the cell? Define three phase concept of the cell. Okay, three phase concept of the eukaryotic and prokaryotes. There are a lot of question. Okay, so we start from the two phase concept. It is very ages ago, and it is not a big thing. So the two phase concept, what it tells? It tells that first consists nucleus and cytoplasm. So the cell consists the two phase concept. So consists of a nucleus and cytoplasm. So the main constituent is the nucleus and the cytoplasm. And the second one is the integrated whole part being considered. So in the two phase concept, we're going to put number 1 marking, that it says two phase concept. So the first phase is consist nucleus and the cytoplasm. The cell consists nucleus and the cytoplasm. And then second, integrated whole part being considered. Okay, so the second one is in the two phase system, the integrated whole part being covered. Okay, the first one in nucleus and cytoplasm. Okay, so the second one is integrated whole part being covered. It means the various organelles are present if it is a eukaryotic cell. For example, nucleoli, mitochondria, Golgi complex, paraplasm. Paraplasm is the collective of ER vacuoles and all. So what it try to sell, the two phase concept sell that cell appeared heterogeneous system. So the cell appear heterogeneous system. What it does it mean? That the first one is it contains of having a nucleus and cytoplasm, and then integrated at whole. This is you have to write. Okay, for integrated at whole means nucleoli, mitochondria, Golgi complex, paraplasm, and you should write in the summary that cell appeared heterogeneous system. So the cell is heterogeneous. It's not a very conceptual thing. It's just a ratta kind of thing. Now we have three phase system. So three phase system, MMG. M means membrane, M means microtubules and fibers, G means granules. So the three phase system talk about the membrane of the cell, microtubules and the fibers of the cell, which provide the structural support and help in cell division. And then we have granules. So MMG, membrane, then we have microtubules and fibers, and then we have granules. So after the discovery of electron microscopy, changed the concept, and that concept lead to the three phase concept. And the three phase concept talk about membrane, then we have microtubules and fibers and granules. So we start from the membrane. So membrane are lipoproteinaceous with or without pore. So first is membrane is lipoproteinaceous with or without pore. So the membrane is lipoproteinaceous, means they are made of lipid and protein in the nature, and they might have pore and without pore. It means the porins may be present or not. For example, in the outer layer of a mitochondrion, the porine receptors was present. Okay. So the second one is polymorphic in nature. So the membrane, yeah, you're right. The plasma membrane is polymorphic in nature. So there is no perfect concrete shape of the plasma membrane. They are polymorphic in nature. They can adapt to the situations very well. um, when it is winter or summer, they can adapt. They have anti-freezing molecules also. Okay, we will talk about later. Okay, so the first one is a lipoproteinaceous structure with or without pores. Then we have polymorphic in nature. They have a polymorphic in nature. The shape is polymorphic. Thickness is tripartite structure. So the thickness of the membrane is tripartite structures, and that is 25 angstrom, 25 angstrom, and 25 angstrom. That is tripartite structure. So the thickness is 75 angstrom. Why 75 angstrom? Because the membrane is tripartite structure and each having 25 angstrom of the thickness. So the overall thickness of the membrane would be a 75 angstrom. A thick which consists of lipid and protein, and each being thick 25 angstrom, and that is tripartite structure. And then we see that semi-permeable or selectively semi-permeable, it means the controlled endocytosis and exocytosis. So what we see is the lipoproteinaceous in nature with or without protein. Then we see that polymorphic, the structure would be a polymorphic, and thickness is tripartite structure, which is 75 angstrom. And then we see the selectively semi-permeable nature of the membrane, which is help in the control endocytosis and exocytosis. And due to universal in structure, the membrane called unit membrane. So why the membrane, plasma membrane called unit membrane? Because it is universal in nature. That is why it called unit membrane, and unit membrane hypothesis came. So what does it mean by the unit membrane hypothesis? This is the question. All else are bullshit and very knowable things. Okay. Now the question is 2 marks question. On basis of high resolution studies, unit membrane divided into 2 groups. So the question could be on the basis of high resolution, microscopic resolution studies, the unit membrane divided into 2 groups. So the first group is asymmetrical structure, and the second group is symmetrical structure. Okay. So we talk about the symmetrical, we talk about the first group, which is asymmetrical structure. And on the basis of high resolution, unit membrane divided into 2. So the first one is asymmetrical structure. It means, what does it mean by structure? So the unit membrane is lipoproteinaceous in nature. So the structure would be the asymmetrical structure, mean the lipoproteinaceous structure would be asymmetry. So asymmetrical structure, it means the inner plasma membrane thicker than the outer plasma membrane. Why? You don't have to remember why outer is thinner. Outer is thinner and inner is thicker, because the outer is in the continuous of the molecular trafficking, and if the road is too thick, if the road is too, there are a lot of vehicles in the road, then it would be a problematic for the molecules to be trafficked on side and off side. So that is why the upper layer in asymmetrical structure, the very first basis on which the unit membrane divided into 2 groups on the higher resolution, asymmetrical structure, it means the upper layer of the plasma membrane is thinner, upper layer of plasma membrane is thicker than the outer protein membrane. So the upper plasma membrane is thicker means that, so for the molecular trafficking, it is selectively semi-permeable. So you need greater amount of force, and that force is thicker, result in the thicker protein in lipid composition. And examples are important. So the first one is asymmetrical. The outer inner plasma membrane thicker than the outer membrane. Okay, inner plasma membrane thicker. So the niche wala thicker and outer wala thinner. That is asymmetrical structure. Contain plasma membrane and vacuolar membrane having thickness 90 to 100 angstrom. That is important. So on the basis of high resolution, we have two groups. So the first one is asymmetrical structure, means the upper layer is thinner, lower layer is thicker in the composition of lipid and protein, and it contains plasma membrane and membrane of the vacuoles. For example, the membrane of the lysosomes, the outer layer is thinner and the inner is thicker. And the second group is G2, include a symmetrical structure. It means symmetrical structure means that both the outer and inner layer having the same thickness and composition, and it includes everything. If you learn one thing, the group 1, which is nuclear membrane and the membrane of the vacuoles, then you don't have to remember this example because those all fall in this category. So the second one is symmetrical structure, means the upper and the lower layer of the membrane have the similar thickness, and they includes Golgi, mitochondria, nuclear membrane, endoplasmic, lysosomal membranes. Okay, so these are the all about the three-phase system, and we covered the membrane. So I'm going to revise this. The first one is lipoproteinaceous nature without pores. The second one is polymorphic in nature. The third one is tripartite structure, each 25 angstrom and together they 75 angstrom. Then we have semi-permeable nature, means the controlled exocytosis and endocytosis mean the molecular trafficking. They are universal, that is why unit membrane. And then we have on the basis of higher resolution two groups. The first one is symmetrical, the second one is asymmetrical. In symmetrical we see the upper layer is thin, lower layer is thicker in the composition, and we saw the examples of plasma membrane and membranes of the vacuoles. And the second one is we see the symmetrical structure, the composition of lipid and protein is roughly the same, and my examples are mitochondria, Golgi apparatus, and lysosomes and other. Okay, then we shifted to microtubules and fibers. Okay, microtubules and fibers, I have to remember things, that is why I'm repeating a lot. Okay, I have to remember so that I can, because these are not very conceptual things, you have to remember things. Okay, you have to remember the points, so it would be easy for the examination. So I was repeating because I was try to memorize the things. Okay, because memorization is also a part of study and it is very important. I do have clarity about the concept, what does it mean by the controlled exocytosis and endocytosis, why it is happening. I do have knowledge, but I must have recalled those points in my paper during my examination so that I can write. Once I recall the points, I can definitely write whatever it takes. So the second one is microtubules and fibers. So microtubules and fibers are rolled membranous structure. So microtubules do have membranous structure. It means they are not in the prokaryotes, because the prokaryotes lack membrane organelle. So the prokaryotes obviously they do not follow mitosis and meiosis, so they do not require those things. Okay, yeah, they do have in their flagella and all, but they have different composition, flagellin. Okay, so they are rolled membranous structure, like a cylindrical structure, as you can see here. Maybe it's not, yeah, here. So it's a cylindrical structure. Fibers are solid, composed of subfibers. So the microtubules and fibers, so the they are the rolled membranous structure, and fibers are solid. So the fibers are solid and composed of subfiber. Obviously the fibers are solid and composed of subfiber. This is important, what I am going to read next. On the functional point of view, in membrane we see on the basis of resolution 2 division was there. Here we see on the functional point of view of fibers or microtubules are divided into two types. So on the functional point of view, the fibers are divided into two groups. The first one is inert. Those fibers who do not show movement, they are help in structural support. They do not forms the movement. Okay, so the inert structural fiber, because they are providing the structure, but the structure is fixed, which is inert structure. They do not required movement. So the inert structure, okay, so it covers inert structure. So inert structure में भी हमें दो structure मिलते हैं. First intracellular inert structural fiber, then extracellular cell के बाहर inert structural fiber. So intracellular inert, so on the functional point of view we have inert structural fiber and active structural fiber. In inert structural fiber we have intracellular inert structural fiber and then we have extracellular inert structural fiber. If we talk about the intracellular कि अंदर सेल के अंदर ऐसे कौन से inert structural fiber हैं, so the chromosomal fibers. So the chromosomal fibers are inert, okay, inert intracellular fiber, okay, so they do not react, okay, they are inert and they are intracellular inert structural fiber. So the chromosomal fiber, the first one is inside the cell, intracellular. So the chromosomal fiber, neurofilament and elastin. So chromosomal fiber, neurofilament and elastin is the intracellular inert structural fiber. And if we talk about the extracellular fiber, which is inert, then we have collagen. Collagen is the most abundant protein in animal, I think. Yeah. So on the functional point of view, we have two inert structural fiber, and in inert we have two, the intracellular and then we have extracellular. In intracellular we have intracellular inert structural fiber, we have chromosomal fiber, neurofilament and elastin, and in extracellular inert structural fiber we have collagen. And if we talk about the active fibers, they are very well known. So if you remember one, the other is understood, that is the smart work. Okay, so the active fibers, they do have some activity, they do have movements. Okay, so the spindle fiber, yeah, in cell division, cilia, flagella, obviously the movement in the medium, viscous medium or any liquid medium they required. Okay, so the spindle fiber, cilia, flagella and myofilaments, because the neurofilaments do not, but the myofilaments help in the contraction and relaxation. So this is important. So I'm going to recall it. So in microtubules and fibers, we see a rolled membranous structure which is made up of subfibers, and on the functional point of view, we have two structural fibers. The first one is inert, the second one is active. In inert, we have intracellular, in which we have chromosomal fiber, neurofilament, elastin, and extracellular we have collagen. And in active structural fiber, we have spindle fiber, cilia, flagella and myofilaments. Okay, then we have granules, and that is going to be the end of three-phase system. In granules, so less attention in beginning of research. So there is a less attention in the beginning of research about the granules, and for example, the granules are peroxisome. So in the beginning of the research, the focus is very less, okay, on the granules. But later it's been discovered that, for example, if I talk about the photorespiration, so photorespiration is a defense mechanism which is been adapted by plants. How it is a defense mechanism? Because we know that trees, the primary producer, obviously produces a lot of oxygen, and oxygen is oxidative, and it increases the entropy. Thus the oxygen which releases from the plants in tremendous amount, they're going to distort the membrane and the very organelle of the plants. Okay, so they're going to damage, because that amount of excess oxygen, if a plant is producing, then it's going to damage their organelle. So the plants come up with a defense mechanism known as photorespiration, in which they divert the metabolic pathway of the oxygen which is releasing too much, and thus they save the plants from the oxidation and entropy, and that is why the life of a plant is too much. Okay, so in the initial days of the science, there is less attention in the granules, and granules are solid, little bit of hollow maybe structure, and most common and well studied granules are ribosome and intramitochondrial granules. Okay, so the most studied and well studied granule, so the most common and well studied granules are ribosomal granules and intramitochondrial granules, the granules around the F1 and F0 particles. So this is all about the three-phase system. The main concept is in the membrane we have, on the basis of higher resolution, two department, asymmetrical and symmetrical example are important, and on the microtubule fiber we have, first one point of view, two inert and active. In inert we have intercellular, which is neurofilament, chromosomal and chromosomal fiber, neurofilament and elastin, collagen. And then we have active, and in granules we have, the most studied and commonly was ribosomal and intermitochondrial granules. So these are the basics, but these three things are important in three-phase system. Then we have category of cell components. So what are the category of cell components? Okay, so brief note under cell concept. So category of cell concept also came under the cell concept. So if the question asks briefly elaborate cell concept, you have to give the description of the two-phase system. Then we have three-phase system, MMG. Then you have to give the category of cell components. Okay, then you also have to give cellular organization. Okay, and that is the end of cell theory. So the category of cell components. So the first, there are three category of cell components. So there are three category of cell components. So the first category is protoplasmic. The second one is alloplasmatic, also known as dynamoplastic organelles, and the third one is elastic, paraplastic or metaplastic. So the first one is protoplasmic, the second one is alloplastic or dynoplastic or dynoplasmatic. Okay, plasmatic है ये, ठीक है. So the first one is protoplasmic, the second one is alloplasmatic or dynamoplasmatic, the third one is elastic, paraplastic or metaplastic. So the first one, if we talk about the components of cell, the first one, protoplasmic. Protoplasmic means the comprises most active particles of the cell. So what are the most active particles of the cell? So indeed we have Golgi apparatus, which is the most active. So in the most active particles of the cell called protoplasmatic, protoplasmic, we have four: the nucleus, the mitochondria, plastid and Golgi. Nucleus, mitochondria, plastid and Golgi. These are the four most active component of the cell, and they falls under the category of protoplasmic. Protoplasmic means they are the most active particles of the cell, and these are the four: nucleus, mitochondria, plastid and Golgi apparatus. The second one is alloplasmatic, or also known as dynamoplasmatic. It means they have some movement, dynamo, dynamic. So in such they include secondary and non-self-dividing bodies. So alloplasmatic means such organelles which are not self-dividing and they are secondary. For example, flagella, cilia, spindle fiber, myofibrils, neurofibrils. So whatever we learn in the microtubules and fibers in inert and active, we're going to give example in alloplasmatic or dynamoplasmatic. They do not have the self-division capability, and they are flagella, cilia, microfibrils, neurofibrils. You can give two examples. Protoplasmic, most active part. Okay. Then we have ergastic, paraplastic or metaplastic. These are the three components of cells. So the ergastic, paraplastic, metaplastic means they are passive type secondary product of protoplasm, which includes secretory, granules and vacuoles. So recently we talk about granules, and these falls into category of that. So the category of cell components under the cell theory, we have protoplasmic. It means the most active part of the cell, which are four. I remember the examples. Then we have alloplasmatic and dynamoplasmatic. They are non-self-dividing and they are secondary. And the third one is ergastic, paraplastic and metaplastic. And these ergastic, paraplastic, metaplastic are passive, and they examples are granules and vacuoles. Um, they are passive type, and secretory granules and vacuoles are the example. Then we have obviously very easy, the cellular organization. The cellular organization, and that is the end of cell concept. This is characterized by the presence of one or more cell in the body. For example, the unicellular and the multicellular. And cellular organization हमने देख लिया. Then we have it organized into complexity. So cellular organization, unicellular and multicellular. Then we see the complexity, which is eukaryotic and prokaryotic. That is the end of cell theory. Okay.